The trial I'll introduce today is based on our finding of PTPRZ1-MET fusion gene. So first, I'll introduce the research progress of PTPRZ1-MET ZM fusion gene. In 2004, CGGA database was prepared to be established by academician Tao Jiang, and the follow-up was started. Full platform sequencing of the second generation genome of whole grade gliomas began in September 2011. Then we obtained vibritiniib's exclusive license patent in China in October 2012 and applied for investigational new drug to CFDA in December 2013. In 2014, CM fusion gene was found to be mainly present in patients with secondary glioblastoma. We published this paper on Genome Rees. Then we constructed the patient-derived cell line and animal model transfected with ZM fusion gene in August 2015. The patent for glioma indication and the clinical research approval was obtained in 2015. In 2016, the abnormal expression of MET was identified with temozolomide resistance, and the phase 1 clinical trial of glioma was launched. In May 2017, ZM fusion gene promotes malignant phenotype and temozolomide resistance through exome secretion. We published this paper on oncogene. Then in 2018, the phase 1 clinical trial of glioma completed, and the progression mechanism of glioma was driven by CM fusion and MET exon skipping, and the results of the phase 1 clinical trial was published on Cell. Then we launched the phase 2/3 clinical trial. In addition to the identification of CM fusion glioma samples, we also identified recurrent CM fusion and high occurrence rate MET exon 14 skipping in brain metastasis in 2022, and the phase 2/3 clinical trial was completed in 2023. So what's ZM fusion gene? We have identified a novel recurrent fusion gene in glioma. The 4 fusion transcripts involved 4 different breakpoints within the PTPRZ1 coding sequence, whereas the breakpoints in the MET gene were exactly the same. The ZM fusion occurs around 50% of patients with IDH mutant astrocytoma, World Health Organization grade 4. Survival analysis demonstrated that IDH mutant astrocytoma, World Health Organization grade 4 patients with ZM fusion had poorer overall survival than those without this fusion. Median overall survival in patients with ZM fusion was 4.2 months, even worse than the overall survival of 5.0 months in recurrent glioblastoma. Next, academician Jiang's team developed a highly selective ATP competitive small molecule MET inhibitor PLB1001, which was also called vibritiniib. In vitro analysis showed that vibritiniib had higher apparent permeability and lower efflux rate than other MET inhibitors. Next, we identified that ZM fusion resulted in hyperactivation of MET and STAT3 signaling. The phase 1 clinical trial of vibritiniib identified RP2D as 300 mg BID and showed well-tolerated and demonstrated preliminary anti-tumor activity of vibritiniib in IDH mutant high-grade glioma. The drug concentration in the cerebrospinal fluid increased with the dose. The CSF concentration in patients treated with 300 mg dosage had reached the corresponding 90% effective dose, AD90, in ZM positive U87 cell line derived xenograft models. This is one of the 2 enrolled patients achieved PR. After radiotherapy and 8 cycles of TMZ treatment, the tumor reoccurrence and the patient was enrolled for 5 cycles of vibritiniib treatment. tumor shrinkage accompanied with symptom relief was observed in this patient after 4 weeks until 16 weeks of ibrutinib treatment. No radiological progression was detected until the week 20. This article has been published on Cell. This is the cover we designed with some are interesting threads in this paper. In Chinese ancient legends, there is a mythical creature named Nian. It often appeared at New Year's Eve and hurt human beings. The only thing that the Nian feared is firecrackers. As time passed, playing firecrackers becomes a Chinese traditional entertainment at every Chinese New Year. In this cover, the mythical creature Nian depicts gliomas with MET alternation, and firecracker depicts the specific MET inhibitor ibrutinib. Cutting-edge research, technology and therapies cure for CNS tumor patients. Joint consultation. Dedicated to providing cutting-edge glioma research, technology and therapies. Cure for gliomas and other central nervous system tumors. Relying on the expertise of academician Tao Jiang's team at the Beijing Tiantan Hospital. Our multidisciplinary diagnosis and treatment MDT clinic is composed of leading physicians from neurosurgery, neuro-oncology, and other specialties. We integrate various cutting-edge treatment methods, including unique domestic and internationally advanced targeted therapies, tumor treating, FILES therapy, and our self-developed TRIS CAR-T, along with other comprehensive treatment plans. Our clinic offers comprehensive support and consultation, as well as personalized treatment assessments and plan development, promoting the application of the latest therapeutic technologies with the aim of improving patient survival rates and quality of life.
Doctors and joint consultation. 陶江、宝石陈、小慧仁、赵石宝。Clinic date every Tuesday morning. Clinic location: second floor International Department, Zone B, Beijing Tiantan Hospital. Address: number 119 South 4th Ring West Road, Fengtai District, Beijing, PR, China.